Last time, we discovered what can happen when the DNA inside our cells becomes damaged. But here's where the story takes an unexpected turn.
Because sometimes the DNA sequence itself hasn't changed. The genetic blueprint is still there, intact.
And yet, two cells carrying the same DNA can behave very differently.
So if the blueprint hasn't changed, what has?
The answer lies in something happening around our DNA, something that helps determine which genetic instructions are used, when they're used, and how strongly they're expressed.
And as those patterns change over time, they can influence how our cells function and potentially how they age.
This is our second hallmark of aging, epigenetic alterations.
And to understand it, we first need to discover how your cells can change the way they read the very same DNA.
So how can the same DNA produce different cellular behavior? One answer is that your cells have sophisticated systems that help regulate how your genetic instructions are used.
Think of them almost like biological dimmer switches. They can influence whether certain genes are more active, less active, or relatively silent, without actually changing the underlying DNA sequence.
One mechanism is DNA methylation, where small chemical tags can influence gene activity.
Another involves histones, proteins that help package DNA. Changes around these proteins can affect how accessible different regions of DNA are to the cellular machinery that reads them.
And that flexibility is essential. It helps cells adapt and respond to changing conditions.
But as we'll see, those regulatory patterns can also change with age. And when they do, the way our cells use their genetic instructions can begin to change as well.
When we say a gene is turned on or off, that's useful shorthand, but biology is usually more nuanced than a simple switch.
Gene activity often works more like a dimmer. Some genes can become more active, others less active, depending on what a particular cell needs at that moment.
And that helps explain something remarkable. A muscle cell, a neuron, and a liver cell can contain essentially the same DNA, yet look and behave completely differently.
The difference is partly in which genetic instructions each cell uses, and how strongly those instructions are expressed.
This ability to fine-tune gene activity allows our cells to specialize, adapt, and respond to the world around us.
And that's important because epigenetic regulation itself isn't a problem. In fact, it's essential to healthy cellular function.
So, epigenetic change itself isn't something we need to fear. In fact, it's happening inside our cells all the time.
Our cells continually adjust gene activity in response to changing conditions. Things like activity, nutrition, stress, recovery, our environment, and even the time of day.
And that flexibility is a good thing. It helps our cells adapt, specialize, and respond appropriately to what's happening around them.
So epigenetic regulation isn't the problem. It's essential biology.
The concern begins when those carefully regulated patterns start becoming disrupted, and those changes persist over time.
And as we age, that's exactly what can begin to happen.
We've seen that epigenetic regulation is normal and essential. But as we get older, those carefully coordinated patterns can begin to drift.
Changes can occur in DNA methylation, in the way DNA is packaged around histones, and in how easily different regions of our genetic information can be accessed.
The DNA sequence itself may still be intact. What's changing is the way those instructions are being managed and used.
And when those regulatory patterns become less precise, genes may be expressed at the wrong level or at the wrong time.
That age-related loss of epigenetic regulation is part of what researchers call epigenetic alterations, our second hallmark of aging.
And that matters because changing how the instructions are read can ultimately change how the cell behaves.
So now we can bring the whole idea together. Your DNA provides the blueprint, but the blueprint alone doesn't determine what the cell actually does.
Epigenetic regulation helps determine which parts of that blueprint are accessible and how strongly particular genes are expressed.
That influences the proteins the cell produces, the signals it responds to, and ultimately, how that cell behaves.
So two cells can carry essentially the same DNA, yet use those instructions very differently.
And that's why epigenetic regulation matters so much. It forms a bridge between the genetic information you carry and the biology your cells actually express.
And when that bridge begins to change with age, the effects may not remain isolated to just one part of the cell.
Now we can see why the hallmarks of aging shouldn't be viewed as separate events.
Genomic instability can influence epigenetic regulation, but the relationship can work in the other direction too. Changes in how DNA is organized and regulated may also affect how well that DNA is protected and maintained.
And those effects can extend beyond DNA. Changes in gene expression can influence proteins, cellular energy, inflammation, communication, and many other processes throughout the cell.
That's why aging is better understood as a network. A change in one biological system can ripple outward and influence others.
And as we move through the series, you'll begin to see that network taking shape, one hallmark at a time.
Because no hallmark truly ages alone.
So far, we've focused on the instructions, our DNA, and the epigenetic systems that help determine how those instructions are used.
But even perfectly regulated instructions are only useful if the cell can carry them out.
And much of that work depends on proteins. Cells must continually make them, fold them into the correct shape, maintain them, and remove them when they're damaged or no longer needed.
When that entire system stays in balance, proteins can perform the thousands of jobs required to keep a cell functioning properly.
But what happens when proteins begin folding incorrectly? Damaged proteins start accumulating, and the cell's cleanup systems can no longer keep up.
That takes us to another critical piece of the aging puzzle, and our third hallmark of aging.
So, we've learned that our DNA is only part of the story. How those genetic instructions are regulated can influence which genes are expressed and, ultimately, how our cells behave.
And because the hallmarks of aging are interconnected, changes in epigenetic regulation don't necessarily remain isolated. They can influence other systems throughout the cell.
But once those genetic instructions have been read, the cell still has to turn them into something functional.
And much of that function depends on proteins. Proteins that must be made correctly, folded into the right shape, maintained, and eventually cleared away when they're damaged or no longer needed.
So, what happens when that delicate balance begins to break down?
That brings us to hallmark number 3, loss of proteostasis.
In our next chapter, we'll discover what happens when the cell's proteins begin to lose their balance.
And if you're enjoying this series, subscribe so you can follow the hallmarks with us. You can also download our free cellular health guide using the link below or call us at 1-888-288-1892 with your questions.